One of the perks of working with Yellowstone samples is a high amount of sampling opportunities. We try to go in, on average, it seems to be about 4 or 5 times a year down to Yellowstone. We have permits for some backcountry hiking that we can do. So we're able to see hot springs, an area of the park that the general public does not have access to. A lot of the samples that we've gotten metagenomes for are what I'm currently working on with my project at the JGI. I first found out about the DOE's Graduate Student Research Fellowship in a newsletter that Montana State would send around. We would get, I think, bi-monthly emails with scholarship and fellowship opportunities, and that's where I first saw it. A major portion of my project has been working with cellulose and looking for cellulose-degrading microbes in the hot springs, as the extreme temperatures and pH conditions of the hot springs are advantageous for the pretreatment process of breaking down plant biomass for eventual biofuel production. After talking with Tanya and some recent publications that she had put out, looking at novel ways of using a hook and bait type strategy to pull out novel cellulose degraders, we identified our joint proposal as kind of an opportunity to expand on the discovery of cellulolytic microbes in hot springs. Working with Tanya has been a great opportunity. And then being a part of the Lawrence Berkeley National Labs, we had access to the NERSC computing. And a lot of my current research that I've done with the JGI has only been possible due to the connection and the access to the Cori supercomputer at the National Lab. A major portion of my proposal had computational work attached to it. So having a shelter in place, although it did disrupt the research that I had planned, we were able to kind of move some of the goals around and then focus more on the computational work. And then once in the summer, when we were allowed to safely return to the lab, I was able to go back for a month and a half or so to finish some of the DNA and RNA extractions that I had planned in my proposal. But then now I'm back at my home, working from home on the more computational aspect of it. So although the shelter in place kind of disrupted the research, I think it still gave me the opportunity to really dive deeper into the computational work, which is, part of the reason I came to the JGI is to build on those skills that I had not previously learned. I would definitely recommend this fellowship to other graduate students. It has given me an awesome opportunity both to network and work with other scientists at the National Lab, but it's also helped me really build upon some of my bioinformatics skills. I kind of came into this fellowship with just a baseline level in R for figure generation, but now working with a ton of the folk here at the JGI, I've really kind of built upon that R knowledge and then started to really learn the whole shell scripting Python aspect of bioinformatics as well. So it's been an awesome opportunity and has really, I think, progressed my PhD studies.